Good evening, good morning, everybody, according to the time and where you are. My name is Professor Abdel Shoair, Egyptian. I'm going to talk about the uses of a special complex, which is ruthenium 3 complex, and some study in vitro and in vivo for some models. Really, this compound is very simple, and what is the most important, it's soluble in water and ethanol. So it's very easy to make a study on this compound. For these reasons, we tried, I think, better to use it for some anti-cancer study in some earlish mice. Okay. So the title of my talk is Synthesis and the Characterizations of Ruthenium 3 Complex containing an amino ethyl benzimidazole and its anti-cancer activity in vitro and in vivo. The aim of this work includes 3 points. Number 1, synthesis of the complex containing this ligand. This ligand, which is called 2-amino-methylbenzene, is very important and have a biological activity. For this reason, we showed this one for coordinations with ruthenium 3 metal. Next time is characterization of this complex by different spectroscopic technique. The third is application of this complex as anti-cancer agent. Why did we use ruthenium 3 complex? I'm sorry, it's complexes here, but it's one complex. Ruthenium 3 complexes are being studied as anti-cancer agent due to their high in vitro bioactivity against many human cell lines. At the same time, ruthenium complex has different oxidation state. It has about 12 oxidation state. This is make its characters is an extraordinary character. Next, comparing to the well-known anti-cancer complex, which is cisplatinum, ruthenium 3 have been shown to have a low kidney toxicity in animal trial. The creations of less toxic and water-soluble ruthenium complexes led to a small amount of this complex entering a preclinical experiment in anti-breast cancer agent. Ruthenium 3 complexes transportation in the body. I would say their affinity for transferrin ion transport protein. The ruthenium 3 complex binds specifically and promoted to your human serum albumin, HSA. Third, a significant part of cancer treatment consists of exploiting 9R-binding moieties, because the most important element which is used for anti-cancer agent, as you know, cisplatinum, which has been made 1971, and nowadays there is some iron complexes. So decided to use ruthenium, which is the same group and periodic table. Transferrin is primarily responsible for ion transport within the body, while albumin and other protein play a less important role. So why would you use ruthenium? Here you can find a reference in 2021 about the applications of ruthenium complexes for some transferrin fluorescence spectroscopy and some molecular docking, which is published in Biometals in 2021, which explain the coordinations and the behavior of the metal with the DNA. Synthesis of the complex. The most important task in this work, very simple synthesis of this compound. It's made just by refluxing of ruthenium chloride with the ligand, which is 2-amino-ethyl benzimidazole. Very simple synthesis. And nowadays, we are looking for the very simple methods in which very easy to use chemicals, especially in water or in ethanol. We did many characterizations for this complex, like microanalysis, magnetic, electrochemical, spectral data indicated the complex is low-spin complex. I'm sure maybe somebody is not interested in inorganic, but this is called a low-spin complex, a stable and octahedrally coordinated to ruthenium 3. I mean, by this, the ligand coordinates to the ruthenium ion octahedrally, okay? This is the proposed structures for our complex, as you see. 2 mole of 2-methyl benzimidazole with ruthenium chloride, refluxing in ethanol with the corresponding complex. It looks like a green precipitate. The most important, I say it again, very soluble in water, very soluble in ethanol, so makes it study very easy. Anti-cancer activity of ruthenium of this complex. Actually, I have an author working with me on anti-cancer study, and this is his work, or even her work, because she's a woman. Her name is Dr. Heba. In vitro study, measurement of cytotoxic activity by MTT assay. The human cancer cell line, colon carcinoma, HC116, and the breast carcinoma, HCF7 cells, were seeded with different concentration of this compound. The lowest IC15, 75 from the curve, and sometimes 18.08 microgram per ml, indicated the anti-cancer potential of our complex. I mean, by this study, the complex have a very good, or even you can say good, anti-cancer activity. Evaluations of cell death smooth apoptosis. The ruthenium complex has a higher apoptosis actions on cancer cell than the necrotic one. I think this is clear from the curve. As you saw, anybody interested, I'm sure will understand this. DNA laser assay. The across gel for DNA damage assay, we have different lane. Lane 1, DNA laser. Lane 2, colon cancer cell. Lane 3, treated colon cancer cell. We find cells showed smears and fragmentation in the end of lane. Lane 4, treated CF7 cells showed the same smears and fragmentation as you see. The third, well, I think this is the slide number 50, DNA laser assay. This indicates that this complex acts on DNA of the cancer cell and degrades it. I mean, this complex decomposes or causes decompositions of cancer cell in DNA, which confirms the apoptosis effect of our compound. In vivo LD50, anti-cancer activity by transplantations of early sites, carcinoma, EAC. This is the abbreviation in females with albumin mice as a model of breast cancer. 6 groups have been made. Group 1, control group, which take only distilled water only, nothing. Group 2, early site carcinoma. Group 3, treated group with low dose. 4, treated group with high dose of this complex. The 5th one, normal mice taking the low dose. The 6th group, normal mice taking the high doses to detect the drug cytotoxicity on animal organs. I think this picture is clear. The untreated, which is infected with a material to cause cancer, it looks like it's very big and effective. The treated one, the body is small. This means this complex is very serious and important in treating this kind of cell. Liver function tests. Significant decrease in both SGP, liver enzyme, and SGOT of the treated group were observed than the Ehrlich group. From the curve, it is clear. Next, while significant increases in the albumin level in the treated group than the Ehrlich one. The albumin levels were significantly decreased than the Ehrlich group. Kidney function tests. There were a significant decrease in the kidney function tests in the treated group than those of the Ehrlich one. Both urea and uric acid level in the treated group reaches the normal levels of the controlled group. This result confirmed that this complex is safe for the kidney and easily to be eliminated from the body. By the last 3 slides, I would just say this complex, synthesis of this complex is very easy. It gives a good result for anti-cancer activity like liver functions, like kidney functions, and this is clear from this curves. Hematological parameters, there was significant increase in both hemoglobin content and red blood cell count in treated group than those of LH1. However, white blood cell count was decreased only in the treated group with low drug doses. Anticancer and antioxidant enzyme, there was also a significant increase in the antioxidant enzyme in the treated group than in the LH1, and it reaches the normal level of the control group. Conclusion. Before reading this one, I would say this, what is the most important in this work? The most important, as I said, this complex is very easy to make it. Two, very soluble in water, very soluble in ethanol. These two reasons make it very easy to make many studies in this compound. Okay, so anyway, I would say all this result suggested that the complex destroys the cancer cell by increasing the apoptosis. Also, this complex captures the reactive oxygen species and decreases the oxidative stress on the organ without harming the normal cell. The cell, likewise, it has a limited toxicity on liver and the kidney cell. Moreover, this complex didn't increase the hematological parameters and has no toxicity on the red blood cell or on the immune system. Thank you for everybody.